Good afternoon, everyone. Hello and welcome. I'm Victor San Pedro, and I'll be your host for today's webinar, LCMS Metabolomics in Motion, Techniques and Tools for Proteomics and Metabolomics Facility, or the PPMF, of the Philippine Genome Center, along with Rainphil Incorporated and Waters Corporation. Today, we explore the dynamic field of metabolomics, where LCMS technologies are driving new frontiers in health, environment, and biosurveillance. Our focus will be on PFAS, the so-called forever chemicals, and how cutting-edge workflows are being applied to monitor exposure and safeguard public health. You'll hear from three distinguished experts who will share practical strategies, innovative tools, and real world applications that highlight the power of metabolomics in addressing urgent global challenges. After all the presentations, we'll open the floor for a live question and answer. Please stay until the end to complete our evaluation form and receive your certificate of completion. Licensed chemists and chemical technicians will also earn 1.5 CPD units for chemistry. Now let's get started, shall we? By the end of today's webinar, we hope you leave with a deeper understanding of cutting-edge metabolomics technologies and how these tools are being harnessed to advance diagnostics, therapeutic development, and systems biology. But before we start, can I call on any representative from Rainfield? Ma'am Jazil, are you here po?
Hi, Sir Vic. So apparently Ma'am Jazz is not here, so I will be the one welcoming you guys for
Yes, ma'am.
this event. So good morning everyone and welcome to our Metabolomics event. Thank you for taking the time to join us today as we delve into the concerning field of PFAS and its growing impact on research, diagnostics, and innovation. We're excited to learn from our speakers, share experiences, and of course, explore new ideas that can help advance our work and better support the communities we serve. So once again, we welcome you and we hope you enjoy the next sessions ahead. Thank you.
Thank you, Sir Joseph, for that wonderful opening speech. And now, before we begin, a quick reminder, please hold your questions until the end of the presentations. But for our first speaker, the question and answer portion will be held after her talk. Our first speaker is Dr. Anna Karen C. Lacerna. Dr. Karen is a chemist and scientific leader with extensive expertise in advanced analytical technologies and metabolomics. She earned her PhD in chemistry from the National University of Singapore and her bachelor's in chemistry, magna cum laude, from the University of the Philippines, Los Baños. She currently serves as the scientific director and laboratory manager of the Central Instrumentation Facility at the De La Salle University, overseeing chromatography, spectroscopy, and NMR laboratories. She is also a specially appointed assistant professor at Ehime University in Japan and a DOST PCHRD long-term Valic scientist. Dr. Lacerda has delivered keynote lectures and webinars on metabolomics applications in health, food, and environmental research, and has published widely on multi-omics approaches, clinical biomarkers, and environmental toxicology. Her work continues to bridge cutting-edge analytical science with pressing challenges in health, sustainability, and environmental safety. Now, please, let's all give a warm welcome with Dr. Ana Karen Lacernas.
Hello, Victor. Okay, can I be heard?
Yes, ma'am, you can be heard po.
Okay ba?
Yes po.
Yeah, okay. Okay, thank you very much, Victor, for the kind introduction. So, good afternoon, everyone. So, I'm Karen from De La Salle University CIF. So, I'm happy to be part of this webinar hosted by PGC, PPMF, Rainfield, and Waters. So if I may be allowed to share my screen. If you can please allow me to share my screen.
Ma'am, you should be able to share your screen now.
Okay. Yan. So once again, good afternoon. It's nice to see that we actually have Jackie here as one of the main speaker. So hello, Jackie. Jackie was
Hey.
my previous lab mate in Singapore. Okay, so today I will be presenting on MS-based metabolomics and chemical risk assessment. As Victor has shared earlier, in some of my work on metabolomics has involved um, environmental studies, no? And so that is, which is what we call mental toxicometabolomics. But this will also be applicable in terms of chemical risk assessment. So that is what we're going to focus on today, and mainly on mass spectrometry-based metabolomics. So the agenda for today, we will cover first an overview of metabolomics, and then we will talk about mass spectrometry-based metabolomics. And afterwards, we will go into two applications of the use of in assessing specific compounds related na rin sa toxicometabolomic applications. And lastly, I will give a brief of an overview to our facility, the DLSU Central Instrumentation Facility. So first, for the overview of metabolomics, so I think you're quite familiar with this poster, no? Or this illustration. So this is something na nung panahon na nag-aaral ako for my PhD, this is actually posted on the wall of my bedroom because I was tracing through the different metabolic pathways of my study. So this is a representation of the different metabolic processes that's occurring in our body. So hindi tayo mabubuhay, we will not survive without all of these different interconnected and complex network of reactions, no? So these will actually be divided into what we call metabolic pathways. So certain parts will be they will be divided into what we call metabolic pathways. Very prominent will be what we call the tricarboxylic acid pathway. Nakikita natin dito, no? And these pathways involve as precursors as end products what we call metabolites, no? So these are just some examples of the metabolites that are found in the tricarboxylic acid cycle pathway. Small molecules that are produced in or by biological processes. Usually, their molecular weight will be in the range of 50 to 2,000 Dalton. There will be those that we call endogenous metabolites, so those that are inherently found in our body, in the biological system. These will comprise your primary metabolites, which are involved in the primary metabolism. You also have those that are secondary metabolites, which are usually produced as a stress response or as a defense mechanism, no? So some examples of primary metabolites will be your phenylalanine, amino acids, as well as your glucose 6-phosphate. You also have as example of secondary metabolites, which are usually found in plants, in microbes, strychnine, which is an alkaloid, no? So this is just one of the examples. You also have exogenous metabolites, no? So for exogenous metabolites, these are metabolites that were produced from metabolites. So they are foreign to the body, but they are being metabolized for the body to be able to excrete them. So for example, you have acetaminophen. So yung ating paracetamol, uh, this will actually be metabolized by the body and this will form a glucuronide, no? So there will be glucuronidation that will be involved in terms of the metabolism of acetaminophen for it to be released from the body. So your metabolites will comprise of those that are inherently present in the body as well as those that are external but are being metabolized by the body. So metabolomics, you're looking at the whole expanse, no? So when we talk about omics, you are referring to the simultaneous measurement and comprehensive analysis of certain molecular levels, no? So you would have, of course, upstream will be your genomics and transcriptomics. You also have your proteomics. Then a bit more downstream will be your glycomics, metabolomics, and lipidomics. So most of these actually involves the use of mass spectrometry, as you will see here in the illustration. Also in genomics, in proteomics, in metabolomics, glycomics, and lipidomics, mass spectrometry is very much used. So as you can see, there will be different transformations and processes from the upstream, from the chromosomes to the DNA. There will be certain modifications involved from the translation as well as in the transcription processes. And these will all be captured in the downstream, in the metabolomics, in the lipids, in the metabolites part. So when we talk about metabolomics, you're looking at what we call the metabolome, no? So this is first coined in 1998 by Oliver et al. When they were measuring the changes in the metabolite concentrations. Subsequently, dumating na yung term na metabolomics, which was coined by Finn et al. And then you also have metabonomics, no? And this was actually coined by Nicholson et al. So basically, for both of them, there are mainly referring to the comprehensive and simultaneous analysis of different metabolites that are found in a biological system, no? So medyo mas focus lang, medyo mas well-defined lang yung part ng metabonomics as it encompasses different factors. But basically, they both involve the comprehensive and simultaneous analysis of measurements of metabolites in order to characterize a particular biological system. So currently, mainly metabolomics na lang yung ginagamit na term. Metabonomics, not so much. So why do we have to take the metabolomics approach? So ito uli yung ating omics, different omics levels kanina. First, if you look at the genome, the proteome, there will be certain, for example, proteins, no, that are similar between different organisms, like for example, in human, in rats, but their functions might differ, no? But if you look at metabolites, glucose in human will also be glucose in pigs or in rats, no? So there is the similarity of metabolites across the different species. So that will be useful if you're trying to understand like how would like the results of a metabolomic study in an animal model could be associated with a metabolomic study or associated with the effects on the human, no? So that will be important. Now, it is also it is also um it is also close to the phenotypic expression. So when you talk about proximity to the phenotypic expression, you have your phenotype here, which is the manifestation of the biological system. And it will also magnify changes. For example, if there are minor changes in the genome, these minor changes will actually parang nagsa-snowball siya, no, in terms of the effects as it goes downstream. So you will be able to magnify small changes in the genes at the metabolite level. So sinasabi nga na ang metabolomics or at the metabolite level, this is the a functional gene expression of a functional gene expression measurement of it's a sorry, yeah, meron sorry about that. Let me just close the sorry. Okay, so going back, so that's why metabolomics is also referred to as functional gene expression. Yung you are able to see changes at the gene expression at the metabolome level. You will also be able to capture the effects of different environmental factors, no? It could health condition, gut microflora, and host interactions, and this will be all better reflected in the metabolite level. Lastly, it is easier to translate metabolites to diagnostic and monitoring devices. So you're quite familiar, no? If you're diabetic, you will be monitoring your glucose levels. You have your home kits or home devices for that, no? Madaling makabili ng mga glucometers to measure glucose. And we also have diagnostic tests in the laboratory for example, for uric acid, for creatinine, for glucose. So those are also metabolites. And that is readily accessible, no, in our diagnostic centers. You would also have your metabolite screening for the screening of newborn screening. As part of the newborn screening, you will also be measuring metabolites if you're trying to identify the presence of metabolic diseases. So as you can see, yeah, metabolites can be easily measured in different manners, no, for the assessment of. So I'm not saying that metabolomics is the only approach that you should take. Just similar to this map wherein you have different layers covering the customers, the streets, the parcels, the elevations. It is only by putting them together that you are able to see the bigger picture, no, or the a better representation of a particular system. So similarly for the omics approach, each omics level will provide a different layer of information, and it is only through a combination of these different layers that we are able to see a better picture or the overall picture. So there are different metabolomic strategies that can be employed. You have what we call targeted metabolomic studies, wherein you're focusing only at specific classes of compounds or specific compounds. You also have what we call the untargeted metabolomics, wherein you're looking at the metabolome as a whole. You're trying to capture as much of the metabolome as possible, no? And you would have two different in terms of samples that you're going to look at. You could be looking at the fingerprint of the particular biological system, wherein you're looking at the components of the, for example, the cell itself or the organism itself. On the other hand, you also have what we call particular biological system. So for example, if you have a cell culture, you will be looking at the metabolites that are being secreted at the cell culture media. So it's only a matter of, do you want to see particular landmarks, specific compounds or classes of compounds, or do you want to see the whole scenery? So that will be the question that you have to address, no, if you want to start a metabolomic study. Now, in metabolomics, there are different platforms that are being used. So you have MS-based metabolomics, you have NMR metabolomics, as well as spectroscopy-based metabolomics. For spectroscopy-based metabolomics, that could be the use of IR, NIR, Raman, etc. But please take note that for the spectroscopy-based metabolomics, since for example, IR, Raman, they do not really have, the signals will be more of bulk absorbances or bulk interactions of your light or radiation with your sample. So it will not specific compounds. So usually this is used for fingerprinting, no? Wherein you just want to have a chemical fingerprint of a particular sample. So it would be useful, for example, if you're looking at quality control of food, or differentiation of different parts of a particular plant, and you're not after the specific compounds, no? So that could be used for those purposes. On the other hand, if you want to know the specific compounds that are involved in the um, that are being analyzed in the study, you would need either mass spectrometry based or NMR approach. So both of these will provide you information that can be associated with specific compounds, no? So we won't go into NMR metabolomics. That will be a topic for a different talk for, if ever. But for now, we will be focusing mainly on MS-based or mass spectrometry-based metabolomics. So for mass spectrometry-based metabolomics, you still have different approaches. You have flow injection or direct injection MS. You have mass spectrometry-based metabolomics, gas chromatography, and you also have capillary electrophoresis mass spectrometry. Mainly for us, we are using these two, no? LCMS, GCMS-based metabolomics. Um, there will be advantages to using this, no? We'll discuss this later. Capillary electrophoresis is not as widely used. There are it allows you to use very small amounts of samples for analysis, but in some cases there will be reproducibility issues, no? Because you're only injecting very, very, very small amounts of sample. So it's not as widely used as compared to these two. Flow injection or direct injection MS, it could be used more for high throughput screening, but the identification of compounds might not be as reliable because if, for example, you have multiple isomers present in your sample, then it will be hard to distinguish them, no? Because they're coming out at the same time. So they're being detected at the same time. So that is where the advantage of LCMS and GCMS comes in. So we will discuss this a bit more in the first. So there is no single analytical technique to cover the whole metabolome. So each of these techniques will cover certain groups of metabolites. For example, for mass spectrometry based, depending on the ionization mode that you employ, you might be covering different kinds of metabolites. Are you analyzing using positive ionization, negative ionization? If you're looking at more volatile compounds, you might be using GCMS. You also have the option to derivatize in order to detect non-volatile compounds. So for example, if you want to analyze amino acids, carbohydrates, and other non-volatile compounds using GCMS, you can still apply derivatization, no? But it will be additional steps. If you don't want those additional steps in terms of derivatization, you have the option to do LCMS analysis. So directly, you can analyze non-volatiles and slightly volatile samples, not so volatile samples, using LCMS. Nagbabago rin in yung detection or analysis depending on the column you use. Kasi syempre, if you're using different columns, medyo yung iba magsasama-sama or magkukumpol at the start kasi to use different columns in order to highlight more specific groups of compounds. So usually it will be one or it could be two or three of these different techniques that you apply in order to cover a more comprehensive coverage of the metabolome. So we go now specifically to mass spectrometry-based metabolomics. So mass spectrometry, it was first devised by Sir J.J. Thomson when he was trying to separate different masses, separate particles of different masses within a beam. So if you're familiar with Sir J.J. Thomson, aside from mass spectrometry, he was credited for discovering, of course, the electron. But aside from that, because of his experiments in discovering electron, he was also able to devise the mass spectrometer. So, mass spectrometry is an important technique for weighing ions in the gas phase. It could be applied to atoms or molecules. And the basic principle will involve the motion of these charged particles or ions will be based on the mass to charge ratio. So to just give you an analogy, if you have a light source, you pass it through a prism, you have the separation of your different colors of light or radiation. For a mass spectrometer, you have your ions that are your molecules or atoms being converted into ions in the ion source, and they will be separated out through the mass analyzer in order for you to get a mass spectra. So, mass spectrometry can be applied for different analytes. So, if you're looking at volatile or non-volatile organic molecules, wherein you can directly analyze them from the sample, you can do desorption or ambient ionization mass spec. If you're looking at atoms or element, you can make use of ICP-MS. If you're looking more for volatile organic compounds and molecular gases, you can use GCMS. And if you're analyzing semi-volatile and non-volatile compounds, usually it is HPLC-MS and CEMS that are used. But across all these different techniques, a very important criteria is they must be ionizable. So if your compound or atom, well, atoms will be ionized in the gaseous state. But if your molecules are not ionizable, so it will be difficult or not possible for them to be analyzed using mass spectrometry. So this will be the main criteria. So in terms of instrumentation, you have this diagram wherein you have a sample source either through flow injection, LCMS, GCMS, or CE, the chromatography or CE part. And then it will go through your inlet. You have a source wherein the ionization occurs, and then it will go to your mass analyzer. So the mass analyzer will filter and also separate out the ions. And then subsequently, you have it detected through, for example, what do you call this? A Faraday tube. I forgot the term. Or some other detector components, and then it you will be able to get the signal through computer system, no, or the software of your computer system. So this will allow you to control the instrument as well as to record your data. Some important terms in mass spectrometry. In mass spectrometry, you will be looking at the resolving power, at the mass accuracy. So when we talk about resolving power or resolution, it is the ability of your mass spec to be able to distinguish one ion from another, no? So if you have a low resolution mass spectrometer, your signal might be something like this, wherein you will not be able to distinguish one ion from another that is within this range of mass to charge value, no? Whereas if you're using a high resolution mass spectrometer, you will have narrower width for your ions, and therefore you will be able to distinguish this ion from another ion that is in this region, no? So aside from the resolution, so if we say high resolution, narrower peaks, better to distinguish different ions. However, that will also have some compromise, no, in terms of sensitivity, because you make your detection very specific to a particular mass to charge value of an ion. In some cases, medyo it might not be good, no? That is associated with a particular analyte. So that is where sometimes it will be useful to make use of a quadruple, pero it will involve other means of like trying to make the detection more selective, no? Because if by using a quadruple at the front, you will actually be able to capture as much ions as possible. And then later on, meron ka nilang additional step in order to make the detection specific to your compound. Now, you also have mass error and mass accuracy. So, for the purposes of identifying the compounds in trying to determine the chemical formula of a particular compound, it will be useful if your mass error is low, no? If you have high mass accuracy. So, usually this is measured by the mass error in ppm, so which is measured as the theoretical mass minus measured mass. So usually for a high resolution mass spectrometry, this would be around 10 ppm for TOF reflectron. If you're looking at more high resolution or ultra high resolution mass spectrometers, you could be looking at error. Monoisotopic mass will be referring to the atomic mass for each atom that is based on the highest abundance isotope for the atom. So in terms of mass spectrometry, you also have the ability to couple two mass analyzers. No, it could be, in this case, triple quadruple, wherein you have two quadruples and then you have a collision cell in the middle, which is they also consider it as a quadruple, but it's not, no. So that's why they count this as 1, 2, 3, triple quadruple. But actually, the quadruple here will be here and here, no. So your quadruple mass analyzers, for this part, will filter the precursor ion, the one that you want to fragment, and then it will be fragmented in the collision cell. And subsequently, you can actually filter with the second quadruple to detect only specific product ion of a particular precursor ion, no? So this is what we call tandem MS, particularly selected reaction monitoring na tandem MS. So that will allow you to have very specific detection. So you also have the option to select specific ions. That is also what we call product ion scan. So this is tandem MS. Now, in terms of different acquisition modes of mass spectrometry, as mentioned earlier, for single mass analyzer, if you have one mass analyzer, it is usually scan, or you can select specific ions for detection. Whereas if you have tandem MS or MSMS, you have more options in terms of acquisition, no? So as I mentioned, you have your product ion scan. Selected reaction monitoring will be selection of specific precursor ions, fragmenting them and detecting specific daughter ions. You also have your neutral loss scan as well as your precursor ion scan. Now, coupling them with chromatography with MS will allow you to have higher sensitivity, higher specificity, and give you more definitive information on your mass and structure. If you have an HPLC DAD, you will only be able to identify these different compounds if you have the analytical standard, no? Because some of them might be absorbing at the same wavelengths, and you will not be able to distinguish them if they are coeluting. On the other hand, at the mass spectra for each of the resolved peaks. So these mass spectra can actually be used, no, to try to identify the compounds present. Because this mass spectrometry data can be used to determine the molecular formula. If you have MSMS, you will also have additional information in terms of what you can use for structural elucidation. Now, you can make use of an targeted approach wherein you're trying to detect more, as much of the metabolites as possible. Usually, this is using a high-resolution mass spectrometer and using scan or data-dependent or independent MSMS. You also have the option to do targeted analysis using multiple reaction monitoring, and that will allow you to detect specifically analytes of target or target analytes from your sample. Now, let's go into applications of metabolomics in chemical risk assessment. So, you can apply metabolomics for a variety of purposes for different kinds of analysis. These are just some of the usual applications, but this is any study wherein you're looking at changes in a biological system, you can actually apply metabolomics approach. So in terms of chemical risk analysis, chemical risk assessment, you are first trying to identify what is the hazard and what will be the dose response assessment. This will allow you to determine the exposure levels that will not result in substantial toxicity. You will also be looking at exposure assessment. wherein you're looking at actual exposures from certain sources. And lastly, you can also make use of you also need risk characterization, wherein you're trying to determine the risk to human or ecological subpopulation. So these are the different components of chemical risk assessment. Now, where can we use metabolomics? No, so usually it is in this two, no, trying to identify the hazard and what will be the dose response what will be the dose response, and in the exposure assessment. So how can we use metabolomics in chemical risk assessment? It is used for the discovery of biomarkers for risk assessment, or what we can also call as effect biomarkers. You can also use it to investigate mechanisms of action of chemicals. You can also use it to address risk analysis challenges, for example, the effect of low dose exposures or chronic exposures, for example. And you can try to link chemical exposures to health effects. Sometimes metabolomics can actually give you insights, or will allow you to gain insights into early um, disturbance of the function of a particular organ, for example, way before any physical manifestation in the organ can occur. So that is one of the advantages of metabolomics. So this is just two examples that I'm going to show. For example, this first study was one that we have done in collaboration with Professor Xavier Comul of Université de Paris, as part of the Université Sorbonne, Paris Cité, and NUS collaboration grant. The student who mainly did the work here is Gideon Lam, a previous student of Professor Samli, from which group I came from. So this involves the toxicological effects of 2378 TCDD on the skeletal muscle of mice during so dioxin includes structurally and chemically related compounds, PCDDs, PCDFs, and some PCBs. This would also refer to sebesodioxin or 2378 TCDD, which is considered to be the most toxic dioxin congener. So they are persistent organic pollutants. They are mainly produced from both anthropogenic as well as possibly natural sources, so from forest fires, from volcanic emissions. They tend to accumulate in fatty tissues due to their lipophilicity, and they are associated with reproductive and developmental issues, and it can affect immune response, hormone regulation, and may cause cancer. So their accumulation in pregnant women can also affect the babies, who will be more vulnerable to exposure since they are underdeveloped. There is also a possibility of metabolic adaptation with early exposures that may provide protection or re-exposure later in life. So yun yung concept of metabolic adaptation, no? Whether an initial exposure through the maternal exposure of the mother can actually result in either to the exposure or an enhancement of the exposure for the baby later on. So in this study, we used GCQ-TOF metabolomics to look at the effects of maternal exposure on young mice. So initially, your pregnant mice were exposed to either nonane or TCDD, and then subsequently, the pups that were born were also exposed to either the control or TCDD, and then subsequently, their skeletal muscle of different parts were analyzed using the metabolomics approach. So there are different metabolites that showed to be significantly perturbed for different parts of the muscles. There were also observed amino acid metabolism perturbation, which could be associated later on with muscle wasting and atrophy. And because there are certain metabolites that were found to be indicative of metabolic adaptation, no, wherein earlier exposure to the TCDD actually resulted in either the enhancement of the effect after another exposure, or to the attenuation of the effect after the exposure of the mice later on. So this is just to give you a very quick scan. So you have your nonane that refers to like control exposure for the mother, and then subsequently nonane was also injected for the young mice. Nonane TCDD refers to like exposure, maternal exposure, and then no maternal exposure for the mom, and TCDD exposure subsequently for the mice, the young mice. You have TCDD exposure for the maternal exposure, and then subsequently control after for the young mice. And then you also have here TCDD exposure for both the maternal exposure as well as the exposure of the young mice. So there are cases wherein, actually, in some cases, there are changes that are mainly associated with the exposure to nin also. But as you can see, there will also be certain metabolite changes that are associated with exposure. For example, metabolic adaptation. In this case, you have exposure to of the maternal exposure. You have the maternal exposure, and then subsequently, after maternal exposure, in this case, you have a lowering of this particular metabolite. No, so this is an indication of possible attenuation of this particular metabolite. Because of the subsequent exposure. And there will be other cases here wherein, for example, in this case, you have the initial exposure, meron kang medyo bumaba yung level of your metabolite, and then subsequently, together with the exposure of the young mice after maternal exposure, you have a heightened level of your metabolite. Almost similar to the levels of control or no maternal exposure. So as you can see, meron kang either attenuation or enhancement or reduction of different metabolites depending on these different kinds of muscles that were exposed. Another study that we did was looking at the metabolism of tri-N-butyl phosphate in earthworm. This is mainly done by my colleagues as well, students during that time, Wang Lei and Shu Lei, which were also part of our group of Professor Sam Lee. So this one will use LCQ-TOF to study the exposure, the accumulation and biotransformation of tri-N-butyl phosphate in earthworm, Prionics excavatus, upon acute exposure using a filter paper contact test and chronic exposure in soil for 28 days. So here, we studied 10 milligram per kilogram and 50 milligram per kilogram levels of TBP concentrations. You can see that for the 50 milligram per kilogram concentration of exposure, you have highly significant decrease in the level of the TBP concentration in the presence of the earthworm. So this indicates the consumption of the TBP by the earthworm. This is representing the levels of the TBP in the earthworms after acute and chronic exposure. Subsequently, they were subjected to metabolomic analysis. These are the different tri-N-butyl phosphate metabolites that were identified from the earthworms. Mainly, we were able to identify these metabolites based on the presence of characteristic ions such as 78 and 80 as well as 98. Which are characteristic of the presence of phosphates. No? So we actually remove those that are associated with the phospholipids. These are mainly the ones associated with your tri-N-butyl phosphate. So there are three not previously reported metabolites that were identified. So these are the MSMS fragmentation annotation of the metabolites such as your ethanol DBP and sulfated ethylene glycol DBP. Then we were able to identify further the phase 1 and phase 2 metabolite transformations of TBP using LCQ-TOF, no? And we find that actually for the subsequent or later metabolic transformations, it is the M7, M8, M2, M3 hydroxylation and phosphorylation that are high, no, with chronic exposure. So to summarize, MS-based metabolomics is useful in studying biological systems in various fields of application. It complements the various tools that are used in toxicological assessment of chemicals, thus also making it useful for chemical risk assessment. And metabolomics can provide important insights on the metabolism and transformation of chemicals after exposure. So lastly, I would just briefly introduce you to our facility, the DLSU Central Instrumentation Facility. So the CIF is actually located at De La Salle University Laguna campus in Biñan, Laguna. So for those who have not yet visited us, I would invite you to visit us in DLSU Laguna campus. So we are under the umbrella of the Office of the Vice President for Research and Innovation. So we are here not only to help elevate the quality of scientific research for DLSU, but we are here, katulad ng PGC, we are here to support the researchers and students of the entire country. So this is our location in Laguna campus. So just to give you a background, if you're familiar with DLSU Taft, our campus is actually 10 times bigger. So this is 50 hectares. And you would expect that most research developments, innovation, and academic programs, new academic programs will actually be offered here in the LSU Laguna campus. For the CIF laboratories, currently we have 7. So we have 3 that are more on chemical analysis. You have your Consolacion Wairagasa NMR laboratory, wherein we have one of only 2 high resolution 600 megahertz NMR in the country. So we also have a chromatography and mass spectrometry lab, wherein I'm the one in charge, wherein we have an LCMS, LCQ TOF, as well as a GCMS. We also have an elemental analysis and spectroscopy laboratory. We have an integrated electron microscopy lab, imaging and cell culture laboratory, molecular biology laboratory, and we also have the fabrication lab and the wind tunnel lab for the engineering laboratories. So we're currently a one-stop shop, no, for different kinds of research needs. So we conduct service analysis, we provide trainings, consultancies, research outsourcings, and we're very much open to collaboration. So please do let us know, no, if you have any interest in availing our services or collaborating with us. So we have a Facebook page if you want to know more about our activities and the services that we offer. We also have some webinars available in our YouTube channel if you would like to learn more about the different instrumentation. And if you have any queries, please email us at cif@dlsu.edu.ph. So I would like to acknowledge, again, thank you to PGC and PPMF for giving me this opportunity to speak here. I would like to acknowledge NUS, University Sorbonne Paris Cité, and MOE Singapore for the support in the research that I have presented. And of course, my current affiliations, LaSalle, De La Salle University, Ehime University, and DOST-PCHRD Balik Scientist Program, who allows me to do the work that I am currently doing. So with that, maraming salamat po.
Thank you, Dr. Lacerna, for that insightful dive into LCMS metabolomics and chemical risk assessment. We surely learned a lot. I know I did. So let's now proceed to the question and answer portion. So to our dear participants, there is a question and answer box. You may enter your question. So we have one here. So from an anonymous attendee, hi Potok, I'm just curious, based on your experience, how can computational chemistry approaches such as quantum chemical calculations or molecular descriptors be integrated with MS-based metabolomic data? In your view, which aspects of the metabolomic results or interpretations can be most improved through this type of computational integration?
Um, I'm not that familiar with molecular descriptors and quantum chemical calculations, but I'm assuming this would be related to computational experiments like molecular docking, looking at how certain compounds can bind with, for example, proteins, and that therefore would affect the function, no? So actually, metabolomics can be used to validate those kinds of experiments, no? So if you, for example, you are looking at a particular chemical, the effect of particular chemical on certain processes, you can actually validate that, knowing na, for example, a particular protein will be involved in a particular metabolic pathway. You can actually try to see, no, how does the different metabolites in that particular pathway are affected with exposure, and that will allow you to validate the results of your molecular experiments or computational experiments. So that's one way that I can think that metabolomics can be used. In terms of which aspects of the metabolomics results or interpretation can be most improved through this type of computational integration, I think it would go more into the interpretation. So from the metabolomics analysis, you get the different metabolites that are differentially expressed, no? So actually, I think if you want to vice versa eh, kasi parang one way is for you to validate the results of the molecular experiments using metabolomics. On the other hand, you can also look at the molecular docking or computation experiments to validate the metabolomic analysis or results. So it can also go that way, wherein you can make use of, you can see from the differentially expressed metabolites, what are the proteins associated with that, see, and then subsequently look at like how does these different proteins are affected by your compounds. So that will help validate as well your metabolomics experiment. So it could be a two-way street for computational and metabolomics. Aside
Thank
from
you,
mice and
Doctor.
earthworms, what other species are being studied in relation to metabolomics? Actually, it can be applied to any kind of species, actually. There are, of course, mice and earthworms will be the more common models. You also have your zebrafish models. Any kind of organism can be used for metabolomic studies. Yeah, are there already existing studies or issues here in the Philippines that may be addressed by MS-based chemical risk analysis? So far, I'm not yet aware of which study, but definitely it can be used for any kind of chemical-based, MS-based, any chemical risk assessment. It can be used for in order to study that. But so far, I'm not yet aware.
Uh, so in the interest of time, isa pang question, pahabol. May habol ba?
Okay, may isang question dito from Limwell. What are the difficulties, limitations in metabolomic studies here in the Philippines, if any? Well, previously, the limitation will be the availability ng instrument, no? But as you can see, currently we already have a growing number of facilities in the Philippines that already have the instruments available, like PGC, CIF. We also have University of San Agustin. There are also facilities in Davao, DMSFI. So nag-i-increase na yung facilities that are available for metabolomic analysis. I think the next thing will be trying to take the more targeted approach naman, no? Because currently, yeah, we're limited with the number of standards that we have per laboratory. So, we don't really, there's not a lot of people doing targeted analysis. Uh, but, yeah, if you want to validate the results of your study, that is the way to go. So, next step will be to try to gain those analytical standards that are needed.
Okay po. Doc, actually, time is up na po. So,
Mm.
for our dear participants, you can still type in your question and answers, and we'll just forward it to Doc Karen. for her to answer. Pwede niyo po actually sagutin, Doc Karen. Gusto niyo dun sa question and answer box. Ayun
Okay,
po.
unfortunately, sorry, I'll be leaving soon because I have
Ayun po.
another meeting. So again, thank you very much.
Sige po. Before you leave, Doc Karen, so we'd just like to award this certificate po. Ayan po. Ayan. So this certificate recognizes Doc Karen's contribution to today's theme, LCMS Metabolomics in Motion, Techniques and Tools for PFAS Biosurveillance, held this November 14th at the Philippine Genome Center, UP Diliman, signed by Dr. Neil Andrew D. Bascos, Program Director of Protein Proteomics and Metabolomics Facility, and Dr. Wendell L. Rivera, our current Executive Director of the Philippine Genome Center. So, Doc Karen, let's take a picture with the certificate. And then po. Ayan. Okay. 1, 2, 3. Then, before Doc Karen leaves, so may I request our organizers and speakers to please turn on your camera. Magrupo to tayo bago umalis si Doc Karen.
Pasensya na po.
Ayan po. Okay. So, 1, 2, 3, smile. Thank you,
Thank you
Doc
very much,
Karen, for
Jackie
obliging
Shino.
us. Well, thank you.
Bye.
Okay, bye po. Okay, now we'll proceed to the next part of our talk. So we have our second speaker. But before we begin, a quick reminder, please hold your questions until the end of all of the presentations. For our next two speakers, the question and answer portion will be held at the end of the program. So our next speaker is Dr. Ian Ken D. Dimzon. So Dr. Ian Ken Dimzon is an associate professor of chemistry at the Ateneo de Manila University and deputy director of the Philippine Institute of Pure and Applied Chemistry, or PPAC. He earned his PhD in environmental analytical chemistry from the University of Amsterdam and has extensive international research experience, including work in Germany on polymer chemistry and PFAS analysis. His expertise spans mass spectrometry, chromatography, and method validation, with a strong focus on environmental contaminants such as PFAS and microplastics. Dr. Dimson has published widely, delivered international trainings on analytical quality assurance, and received recognition from the National Academy of Science and Technology for his outstanding scientific contributions. Today, he brings his deep experience in analytical chemistry and environmental monitoring to our discussion. So let's all give a warm welcome to Dr. Ian Ken D. Dimson.
Thank you, Victor, for the introduction. And good afternoon, everyone. So please bear with me in the next 30 minutes because I will be discussing a little bit complex of a problem like PFAS. So allow me to share my screen first. Let me just check if I can share. Yeah. Okay. So I titled my presentation as like this: Environmental Detective Multi-Technique Approach to the PFAS Analytical Chemistry Challenge. So I'm Ian Ken Dimzon from Ateneo. This is just some sort of an introduction. The emergence of the anthrosphere has altered the delicate balance of the natural world. And the relationship between chemistry and the environment is known to all. However, okay, we see that there are some changes in this balance. And so this discussion will highlight the detective role of chemistry. So let me begin my discussion to discuss about this movie, Dark Waters. It is a Hollywood film that tells about the real life story of Rob Vila. He's a lawyer who had an experience with the chemical giant DuPont. And the legal cases had led to the discovery of a very known PFAS today, which we call PFOA, or perfluorooctanoic acid, or the C8, as referred to in the film. So PFOA has opened the door for us to see the harm that an industry can bring. The real intention of having the fluorinated polymers were to have some new materials, but then later on, it was found out that they harm the environment. So now PFAS is in many different products, from water-resistant clothing to paints to non-stick cookware to even cosmetics, pesticides, etc. So think of anything, and there's probably PFAS in those products. And PFAS has proliferated in the environment in many different forms. Today, we cannot really talk about PFAS and generalize what PFAS is, except to define what is the feature that makes a compound PFAS. And we associate PFAS with forever chemicals. Forever chemicals because PFOA and its other, let's say, another compound similar to that, the PFOS, are known to be not really degrading fast in the environment. It takes years for them to be degraded in whatever way, whether it's chemical or physical or even biological processes. And this PFAS, represented by PFOA and PFOS, have known effects on human health, as can be seen here in the slides. We can see some effects like cancer, damage in the liver, and even sometimes lead to some inflammatory diseases. And not only that, PFAS is in the environment and they tend to be bioaccumulative, so that when an organism ingests PFAS, they tend to stay in the organism for a longer time. Thus, as they accumulate, they become more harmful or toxic to the organism. So here are some representative forever chemicals. But what is PFAS now? Actually, we have a second definition of PFAS that was just released in 2021 by the Organization for Economic Development. PFAS now is defined as fluorinated substances that contain at least one fully fluorinated methyl or methylene carbon atoms, with a few noted exceptions. Any chemical with at least a perfluorinated methyl group, like this one, CF3, or a perfluorinated methylene group is already considered PFAS. And these are some of those chemicals that can be considered PFAS based on that definition. The new definition is important because later on, when we try to find them in various matrices in the environment or in organisms, we look for this important criteria, whether we detect them using mass spectrometry or NMR or whatever detection technology we have. But how do we distinguish PFAS from non-PFAS molecules, especially in untargeted analysis? So that's what we will tackle later on. But this brings us again to the question or to the claim earlier that PFAS is forever chemical. In fact, PFAS is not forever chemicals, only some of them, and maybe a very, uh, a very few fraction now that we consider forever chemicals. You see, based on the classification of Bach et al., these are PFASs. It's composed of the perfluorinated molecules or the polyfluorinated molecules or the polymers, the fluoropolymers. And the legacy PFASs, we call them legacy PFASs. These are the ones that are actually considered forever chemicals. And their feature is that they have a long chain of fully fluorinated carbon. And where are they coming from? Why are they in the industry? They are synthesized via the telomerization process. So from tetrafluoroethylene and some reaction with pentafluoro and then iodoethane, okay, to form various perfluorinated compounds. So these are the legacy PFASs, which are actually the most important and are well studied now and shown to be what we call forever chemicals with harmful effects on humans and in the environment. Now, legacy PFAS are determined with high reliability using targeted techniques. So we use now the things that are done in metabolomics because metabolomics is a well-established science using various tools to identify and quantify molecules within the metabolome. So we expand this concept to the PFAS universe. And so one of those techniques in metabolomics, also discussed earlier, is targeted techniques. So this is an example of a scheme developed to determine legacy PFASs in wastewater treatment plant influents and effluents. And this is to monitor them for regulatory purposes. You can see that there's no single targeted technique that can cover all the known PFASs. So we have the volatile PFASs, so we have to analyze them using GCMS. But the non-volatile ones are usually covered by LCMS. LCMS or LC tandem mass spectrometry. Now, even with the LCMS, it cannot cover all the non-volatile PFASs. You still have to be specific in terms of the method development to target whether the non-ionic PFASs versus the anionic and the cationic PFASs. So this is an example of an LCMS-MS method to determine before in LCMS, we have what we call the multiple reaction monitoring. So MSMS means you have two mass spectrometers acting as mass analyzers, and in the middle, there's a collision-induced dissociation compartment. So what we do is we choose an ion that is associated with our target molecule. Usually, it's the parent molecular ion, or usually also we can have the, let's say, the protonated adduct of the molecule, and then we direct this ion into the CID and try to fragment that. So for example, for PFOA, we are interested with the M/Z413, that is the deprotonated adduct of PFOA, or that's simply the perfluorooctanoate ion. And then you subject it to the CID, it begins to fragment by removing the carbon dioxide from it, giving you another ion with a mass 369, and that's the one that you actually analyze. So with targeted analysis, we can perform targeted analysis on various matrices and so on and so forth to determine these specific compounds. For example, in our group here in the Ateneo, we are particularly interested with the PFOA that is present in various environmental compartments, such as, for example, the lakes and the dams, etc. And this is what we found. We found some PFOA and PFHXA, which is actually the C6 equivalent of PFOA, in various concentrations around Laguna Lake and then Angat Dam and the Mesa Dam. So, and these lakes are important to us because they are our source of drinking water, for example. So we also tried to determine PFOA and PFHXA in various water refilling stations here in Quezon City. And we found some concentrations of PFOA and PFHXA in those compartments. But the US National Standard for Drinking Water says that the minimum allowable concentration of PFOA and PFOS is actually 4 nanogram per liter. So we are more or less at around those standards. So nothing to worry. We can use targeted analysis to develop technologies to remediate PFAS. So for example, another student was able to develop a material based on this is titanium oxide and MOF, metal organic framework, to remove PFAS from various water. And we also saw PFOA in various packaging here in Metro Manila. But those are PFASs. Those PFASs are from traditional sources. That's why we say that they are the most important PFAS earlier. There is now an alternative chemistry going on because we want to replace those legacy PFASs. So for example, you can see here that this is a legacy PFAS, ammonium perfluorooctanoate. And we try to replace them with ammonium 4,8-dioxa 3H perfluorononanoate, which is actually just almost the same as the legacy PFAS, only that there is a break. So it's not a continuous perfluorinated compound. And at those breaks, we are supposed to have degradation, etc. So this brings us to another problem, okay? So, and also because the chemical industry is highly proprietary, leading us to a lot of many different products now, okay? So in fact, there's an identification gap. There are 6 million PFAS compiled, PFAS substances compiled in various databases, but only 700 are structurally defined compounds. And the others are just there. They are, of course, they are in the databases because they are used in formulations. But when they are no longer used, they end up in the environment. So all of the 6 million PFAS compounds, different in terms of their structures and properties, they will all end up in our different environmental compartments. Thus call for better analysis of PFAS, okay? So that's where non-targeted analysis comes in. So that is mainly for the novel and replacement PFAS. Here is a guide for the NTA of PFAS using high-resolution mass spectrometry. We begin with planning which ionization mode, which MS source, what are the settings. We cannot really target and we cannot really ionize everything at once. So we have to perform multiple batches. Then comes the data acquisition. We will get a large data set. So data reduction is the go-to process in this case. And then we have data evaluation and then data storage. Just imagine a spectra a mass spectrum with so many different peaks, where should you go first, and what to identify first in all of those things? So it's good that we have some telltale characteristics of PFAS that we can use to eliminate some things that are not PFAS. The first thing is the mass defects. As you can see here, these are some PFASs that belong to a certain group. So PFOA, PFHXA, PFBA are carboxylic acid. They all have the same mass defects in the end. We call that the Kendrick's mass defects. And also the sulfonate PFASs have also the same Kendrick mass defects. So by calculating all of these mass defects and plotting them, we can actually pinpoint what are the groups of PFASs that are already present in a large data set. And then we try to detect some repeating subunits and also detect some homologous series for detection. And then afterwards, there are some diagnostic ions and fragments that we can also try to locate so that we can somehow detect or tell that there's PFAS in there. Okay? However, we see that it doesn't lead us to one structure or to the full identification. So the HRMS, high resolution mass spectrometry, even if it is really very, very high resolution already, there are still some limitations with regards to accurate mass, and then the accurate mass versus the mass range of the instrument, the availability of accurate mass fragments and suitable reference databases, and also the lack of standards. As you can see, we have 5 levels of confidence in identifying compounds. And the level 5 is when we have already the full confidence that this is the compound for a particular substance that we see, okay, in our analysis. From level 1, we have only chromatographic information. Then we have expert characterization. Level 2, so we try to input what we know about a certain group of substances to narrow down the possibilities of what the compound is. And then until we have some structures from MS and MS data. And then lastly, we have some HRMS data to complement everything. So it's really is a combination of multiple techniques in order to arrive at identifying a single compound. But then remember, we have 6 million PFASs that we need to identify. And that is also one big problem in the end. So here's an illustration on how we can know a certain group of compound. So for example, we have an unknown fluoropolymer. We first do get the direct mass spectrum, that means without chromatography of that, using, more importantly, high resolution mass spectrometry. And we can study the differences between the peaks to see patterns of homologous series of monomer units, etc., because this is a polymer. Then we do the mass defect analysis, like I discussed earlier, to determine again to confirm the presence of those homologous series. Then we do chromatography to separate them and see which are related to each other in terms of the chromatographic separation and chromatographic properties. And then afterwards, we do MSMS to elucidate the structure, not just MSMS, but MS to the N, preferably with the MS having a high resolution. And then by putting together all of this information, we can come up with our own structural information that we can hypothesize to be the unknown polymer. So it really is a combination of multiple techniques in order to arrive at an identification of a single data or a single compound in that huge mass spec data. And then aside from the identification problem, we want to know whether we are able to cover all the PFASs that are present in or that we are exposed to. Okay, so we do the mass balance, and when we do the mass balance, we notice that the masses do not balance. So this is an example the diagram is an example of a micro mass balance. We look at one environment, and that is the wastewater treatment plant environment, for example. We try to determine legacy PFASs in the influent and legacy PFASs in the effluent. So our hypothesis is that when you measure in the influent, this legacy PFAS should have high concentration, but in the, let's say, in the wastewater treatment facility, they should decrease in their concentration. But what happens is that in the effluent, we see more. So therefore, we say that, maybe there are some sources further of these compounds, so that are in the environment. So we say that, for example, maybe there are other sources, okay, that serves as precursor to the legacy PFAS. It's the legacy PFAS that we want to avoid initially, because those are the main source of the problems now. So the mass balance, there are two directions for this. There's the apparent production of PFAS from wastewater treatment plants, and so there's no mass balance. And second, that the total fluorine counted as fluoride ion is much more than fluorine from determined PFAS in the sample. So much more means there are other sources of fluoride ions initially in the samples that we are testing that we cannot account for, and those are probably PFAS molecules. So we go to some other partially selective analytical methods. So we use all the methodologies that we have, not just the very accurate ones, very selective ones, very sensitive ones, even the non-selective ones we need to use. For example, pyrolysis ion chromatography and graphite furnace molecular absorption spectrophotometry to get us a picture of how many fluoride ions are there in terms of their concentration. And we can link that, of course, the fluoride ion could be coming from inorganic fluoride and also from organic fluoride that are PFASs also, okay? So those are some things that we can do. And then also fluorine NMR can be done in order to determine, to differentiate between organic fluorine compounds that are not PFAS and the PFAS compounds. So in that case, we can have some picture of the problem in the end. So we ask in the end, are there solutions to the PFAS problem? There is solution, but the solution is dependent on how much do we know, okay? And that is where analytical chemistry comes in. So here we have a PFAS toolkit for innovating replacements. There are also some information about human exposure, environment, waste infrastructure, consumer products, industry, etc. So these databases are there. We have to fill in the compounds. We have to fill in the information, and that is only possible if we utilize a variety of techniques, mass spectrometry, targeted or non-targeted ion chromatography, and even nuclear magnetic resonance in order to have the problem solved. So there are proposed alternatives and solutions to the PFAS problem, but there is a large data gap that slows down the process of proceeding further. So I think that ends my presentation. Thank you.
Thank you, Dr. Dimson. That was really a very deep dive into the current landscape of PFAS research, and that is very relevant to all of us right now. So let's take a 10-minute break. Feel free to stretch, grab a drink, and enjoy the videos from our co-hosts.
Can I do the share screen? All right, now I can share my screen. I'm not sure if the audience could hear me because I think previously when the host was introducing, I definitely couldn't hear. So please allow me to do a quick introduction of myself. Mabuhay. Hello, everyone. My name is Jackie, and I am the Food and Environmental Market Development Manager from Water Pacific. Yes, you see it right. My full name is Jackie. It's very unusual for Filipinos, isn't it? Because I'm from Indonesia. So if we are meeting for the first time, nice knowing you. My full name is just Jackie. And today I'm very happy to be here to present to you about our advanced LCMS strategies for PFAS detection and visualization across the complex matrices. So simply put, earlier on, Dr. Ian talked about PFAS very clearly, and then Dr. Karen, she talked about metabolomics. So in this talk, I thought, why not I marry them together and allow us to look at PFAS in metabolomes. So let's map the invisible forever chemical together. Now in this presentation, we have a few agenda, but time is limited, so I'm not going to take too long to do this. And so let's start very quickly with the PFAS global regulatory landscape. Dr. Ye has given a very clear introduction about PFAS, so I'm not going to dwell into too much detail, but I think one of the most important key takeaways from the PFAS is that it is definitely a very important concern. In fact, there are now more than 6,000 compounds that is categorized under the PFAS. There are 6 categories which are now being regulated, which you can see in the table, in this red square, PFOA, PFOS, GenX, PFBS, PFNA, and PFHSS. But there are also compounds which are gaining attention every day, and there are also new discovery that is made every day. So we want to be able to focus on not just the discovery work, but also in those that are regulated. So I think Dr. Ian ended his talk very nicely about whether there are solutions about this PFAS analysis. And the answer is yes. When it comes to waters, we have that solution. Because waters have been working on PFAS for more than 20 years. It's been a long time. Of course, the regulations come very recently, so we can see that whether it is from US or whether it is from Europe, we start to see the regulations for PFAS being released. It started with mostly drinking water, and then now we have food and feed, and even for all users of PFAS. So whether it is food packaging or any food contact materials such as cooking pans, because Teflon is actually a very well-used coating for cooking pots, right? Even for clothes. So there are more and more regulations being released. Now, of course, this in our region, the ASEAN region, we don't really see much regulations yet. The closest one we do see is from Australia and also from Japan. But globally, this is a very growing concern. Everyone is very concerned about the presence of PFAS because they are everywhere. Of course, now we know it is not forever, but it will last a long time with a very long lifetime, half-lives. So if we want to analyze them, it can be quite challenging. So with more than 20 years of experience, Waters can provide you with the best solution because we are so well, I wanted to say experience, but we also have all the different know-how with the right people to have established this analytical solution. So if we want to put it simply, we can reduce it to 5 steps. We have been working with various key regulatory bodies, such as the US EPA or EURL, and that's where we will be able to bring you to stay with you at the forefront when it comes to PFAS analysis. So let's quickly dive into the routine analysis. Now, routine quantitation is for those which are regulated because we already know what they are. These are known PFAS and we want to routinely analyze them. So if I want to simplify it to 5 steps for you, the first one would definitely be sample preparation. I'm not sure if you were watching just the advertisement before this talk. My colleague, Dr. Orgatini, was talking about this sample preparation tool, Oasis Wax. Yes, this is our very own SPE cartridge that is highly sought after to perform PFAS enrichment, not just extraction, but also pre-concentration, so that we can perform very trace level concentration, trace level quantitation of PFAS. Now, wax comes from the word weakly anionic exchange. So it comes with 2 different portions where the blue part is allowing us to bind to these hydrophobic perfluoro groups. Whereas we also have this end, which is weakly anionic, that will allow us to bind to the COO tail of our PFAS compounds. So whether it is a long chain or a short chain like GenX, which can be more easily break down than the other PFAS compounds, we can trap them and we can pre-concentrate them. Then after we do the sample prep, we now want to inject it into a system for analysis. One thing that we want to take note about PFAS is that most of the time when we are in the laboratory, the things that we are using are actually plastic. Think about the pipette tip that you are using, or even the vials. You know, in the vial, there is a cover, the cap. The cap is lined with Teflon so that it remains inert. But that means it will actually create more false positives to our samples, isn't it? So what we do is we change the vial, the septum, and also we recommend using only glass pipettes. And in addition, we also have the PFAS kit. The Waters PFAS kit will reduce the contamination in the system. So, for example, we use the pig tubing, and then we also use various columns that's designed to be minimizing contamination of PFAS. And at the same time, we also have this delay column that is critical when it comes to separating unwanted contamination peak from the target PFAS compounds that we are looking at. So you can see that usually, in matrix, there's so many things, especially when you're talking about metabolomics, then you have so much more complex matrices. Then we want to use delay column to separate out all these compounds and even other PFAS from those that we are interested in so that we can quantify them easily. Now, step 3 is the chromatography. Well, we have this wonderful column. The Waters Premium Max Peak series comes with a very special coating, the high performance surface. This will reduce any non-specific adsorption. So what happens is, after you enrich all the PFAS, supposedly they are, of course, trace level. And then now they enter the system. We have contamination kits, so they are passing through the system nicely. But once they reach the column is made of metal, right? The surface is metallic. There will always be certain van der Waals interaction or even ionic interaction that will bind these compounds that we are interested in to the metal. Now we lose the sample, we lose the target analytes. That is not what we want. So by using the max peak columns, we have this special coating that allows us to have much better passing of the PFAS compounds through the column. And thus we improve the analyte recovery, sensitivity, and even reproducibility. So you can see the results over here. Every peak is very visible, very sharp, easy for quantitation. And step 4, we will analyze them by a mass spectrometer. So I think Dr. Karen mentioned about different types of MS earlier, And for targeted analysis, we typically want to use TQ, which is also known as LC-MS-MS. The reason is by having this MRM, we can target these compounds very easily. So we can go down to much lower concentration limit than what is being listed in the regulation. So you can see from the US EPA regulation, the lowest amount of PFAS is supposed to be monitored at 4 ppt. But with our MS TQ Absolute XR, we can go down to an average of 1 ppt. That's no wonder why our LCMS has been used by a lot of different regulatory bodies. And we are very proud to say that many of these methods were developed on our system. So we have all the methods, the know-how of handling the PFAS. I'm not kidding. And in this example, I wanted to showcase one particular example where we are doing PFAS in water. Now, PFAS in water, by right, we are supposed to do pre-concentration, right? But our instrument is so sensitive that what we did is only filter and inject. Yes, just filter and inject. So in fact, in this case, we don't need to use the SPE to enrich the PFAS because the instrument is so sensitive. We can just inject them directly into the LC and MS for analysis, and then we will be able to get this kind of sensitivity. Now, of course, this is an ideal case, a very clean sample matrix where it is water. And when it comes to like biological sample matrix, definitely step 1, sample prep is still very important, yeah? Then step 5, after obtaining all the data, I think one of the most important things is to analyze the data. So we have our software, Waters Connect, which is designed for quantitation. Yes, it works perfectly with our TQ, and it even has unique functions that is designed for PFAS reporting. So for example, if we are following certain US EPA methods, they will have specific requirements such as the RSD report or the internal standard recovery. Now, rather than making it one by one for your own report, we already have the template ready, and you just need to load the template, and you can get those results being reported according to the regulatory requirements. So this allows us to easily and confidently submit the reports for PFAS quantitation. Now, if we are not really looking at just quantitation, because quantitation is targeted, right? What if we want to do discovery work? We want to look at PFAS, which are still unknown to us, novel PFAS. We actually have the LCMS tools as well. This is where I would like to introduce you high-res MS, which Dr. Ian mentioned as well. Now, because we have heard many reports over the world. We have heard about how PFAS is found in human blood, right? I'm sure you have heard also about PFAS being found in human milk, the breast milk, and then the polar bears. In fact, I heard some rumor saying that Philippines has a lot of PFAS around the area. So we want to confirm that. And how do we do that? We need to do more discovery work. Targeted PFAS, we can do those which are already known. And for those unknown, we want to do non-targeted screening. We will need high-resolution mass spectrometers. So this can be useful for us to discover novel or emerging PFAS, or if we want to find out the precursors, or even the transformation product, if we want to identify them from a very highly complex matrix, especially when it comes to metabolomics, or even if we are interested to do spatial or molecular imaging. This can be very crucial, and high-res MS is actually the way to go. High-res MS, however, is something that is rather difficult to use. I mean, if you have done research on high-res, you would know it is very complex. It's not just the sample matrix is complex, the data that we obtain is very difficult to analyze. So we want to talk about, let's say, PFAS which are unknown. These are normally, when used, it is not disclosed in the first place by the producers. Then how are we going to identify these compounds? So we have to be able to find them without a reference. And so when we are trying to report them, we need to have enough confidence levels. So how can we be confidently telling the people based on the report that you can be confident? So if you have seen the example previously where Dr. Ian was doing the research, he had to look at multiple references, mass defects, characteristic product ions, all the telltale characteristics. So it's a very time-consuming process. And when it comes to limited resource, it's what is happening in every single laboratory. So if we want to be able to optimize the entire workflow, how can we help you? You are asking, right? Okay, I tell you. Now, so Waters, we have a lot of high resolution mass spectrometer, and they are designed for different applications. Particularly now, if we are looking at the unknown PFAS screening, I would love to introduce to you to our Select Series Cyclic IMS, which is designed for discovery and research. So it comes with the highest ion mobility resolution, and it allows multiple passes through the ion mobility cell. So this will allow unprecedented separation power. And this is particularly useful for advanced structural analysis and even isomer characterization. So it is deeply connected to omics study, and this is most relevant to you, in my opinion. And at the same time, we have multiple ion sources. So one of the ion source that is most relevant and most interesting to you might be DESI, because this allows us to do imaging. But let's say we also have a lot of other kinds of ionized ion source, such as the ASAP, which is known as the Atmospheric Solids Analysis Probe, allowing it to analyze directly from solid sample, or even to use GC. Yes, we have the APGC to connect to our LCMS. So this allows LCMS to also analyze compounds which are non-polar and also volatile. This works also for high-resolution MS. And I think most importantly is this software known as the pattern analysis. This is our sample characterization software that will help you to confidently and flexibly identify compounds, especially such as PFAS. So I'll show you an example of how it works. It is actually very user-friendly. So if you remember the presentation from Dr. Ian, he has shown you the mass defects and also, like when you're trying to find certain products, because HF, for example, will be eliminated from the collision cell when it is being produced. So HF will be 20. So anything that will produce 20 will have the same mass defect, will likely be the PFAS. And of course, the more information that we have, the better it is for us to filter and visualize. We can even combine it with CCS information, which is known as a collision cross-section. Collision cross-section is something that is very helpful to combine with ion mobility, because whether it is a linear or branch, the shape of the molecule actually decides the ion mobility. So this is something very unique from Waters, but can be very helpful for you to identify and do all these small molecule workflows. So this will reduce complexity significantly and increase the speed to results, because we can also showcase the data in these dynamic interactive tables and graphs. So I will show them to you in two different examples. Let's do the visualization of PFAS across complex matrices. So in these two case studies, the first one I'm going to show you is about how we can use the cyclic MS, cyclic ion mobility mass spectrometer to perform the human serum study. So by combining with the holistic data workflow of pattern analysis, we are trying to analyze the human serum that are from e-waste handlers. So when people are handling the electronic waste, we are trying to see whether they are being exposed to PFAS. And what we can do is to apply that pattern analysis software. Now, so in a high resolution MS, data can be very complex. So especially if you are performing data independent analysis, DIA, you can get like tens of thousands of components. So which of these compounds are actually the novel or emerging PFAS compounds, right? And also, if we are looking at compounds which haven't already been registered somewhere, there's no library, there's no standard, how do we enable the putative identification of these known and unknown PFAS? What is putative, you ask? Yes, so like when you don't have the library, we don't have the standard, we want to identify them. With enough information, we can conclude that the compound is dead, but there's still no proof, so we call it putative identification. Now, so this is how the interface will look like, and this is for the PFAS sample. And what we do is by applying different filters, like the Kendrick mass defects and then the the characteristics, telltale signs, you can then identify the compounds, and then we can visualize from the map of data which are the suspected PFAS. And then from there, we can do a library match by pattern. So it is not a full match, but like when we match it against a library, something along the line of 50% will mean that it is much more likely than 0% to be the compound that we are interested in. So that relevance and the correlation will allow us to putatively identify the unknown compounds. And then in this example, we found the structure putatively of these 4 PFAS compounds. So this is how the pattern analysis software can be extremely helpful for your high resolution MS analysis. And then in my second case study, we are trying to image PFAS across matrices by using DESI. Now, DESI is the desorption electrospray ionization. So what it does is when you have sample, you put it in, you can do imaging. You can be coupled with both the TQ and also the high-res MS. So if you remember, TQ, usually we use it for targeted quantitation, and then the high-res MS, we are using it for unknown discovery. But in either case, we can use the DESI to perform the imaging. So it has the advantage, both sides, because TQ, we are targeting specific MRM. So we can attain very good sensitivity. Whereas for high resolution MS, we have the discovery flexibility. So in an example where we are trying to analyze the porcine liver, yes, liver of baboy, we are analyzing and we spike them with the PFAS mixture. So you can see from these spots, when it gets higher concentration, the spots get brighter. So it is easy for us to do any imaging. Like you can literally visually see them. And because of the different combination of MS, we can go down to detection of 10 nanogram per milliliter or 10 ppb parts per billion. So this is a very powerful technique. Not just can we see them, but we can identify them. And in a case where we are trying to do lentil analysis, so we grow the lentil in PFAS solution. So on purpose, we are trying to contaminate the lentils with PFAS because we want to simulate the real world, right? If the water is highly contaminated with PFAS, how would plants take up the water and thus getting PFAS stuck in them? We can actually see these images over here. You can see the red line inside the xylem. You can have yellow spots. Yes, the bright spots come from the presence of PFAS. We can actually localize where the PFAS is being distributed. We know how it is being spread within a certain organism. And because cyclic ion mobility MS allows us to even check between different conformation, whether it is linear or it is branch, you can use this to study whether which one is more dangerous for the intake of living organisms and so on. So imaging can be a very powerful tool to study the biological uptake of PFAS. Now, so we want to wrap up this presentation. I would like to share with you, like from Waters, we have a whole bunch of solutions for PFAS analysis. Most important thing that we always do in the laboratory is to have sample preparation. So sample preparation will enhance extraction recovery, enrich the sample for us to do trace quantitation. And if you're interested to do automation because you have a very high amount of sample to analyze, we have such option to allow you to do automated sample preparation for the extraction of PFAS. And of course, we also have the PFAS kit, which I highly recommend you. When it comes to PFAS analysis, we really need to make sure that we minimize the contamination of PFAS within the system. So when we are reporting any PFAS, we can be confident that it does not come from the instrument, but it really comes from the sample that we are trying to analyze because this is so prone to contamination. And then with 2 different types of MS, we have the TQ LC-MS that has the MRM function, which offers the highest sensitivity for routine quantitation of known PFAS. And we have cyclic IMS, which is a high resolution MS that allows us to, it has reduced chemical noise function, and it can help us to differentiate even PFAS conformers when it comes to unknown PFAS identification. A software like pattern analysis is someone that facilitates a holistic approach to discriminate unknown PFAS from tens of thousands of metric components, even in DIA. And we also have the DESI access, which enables visualization and direct detection on tissues without extraction. Yes, I didn't mention this, but like the porcine liver cross-section, we can just place it into the DESI, and then we can do the visualization directly. With more than 20 years of experience handling PFAS, Waters is the analytical partner that you can stay with when it comes to PFAS analysis. We know whether you are doing routine quantitation or you are doing discovery. We have all sorts of solutions from sample prep, contamination management, chromatography, mass spectrometry, and data analysis for your PFAS analysis. So if you like my presentation and if you would like to find out more, please feel free to scan this QR code and you can leave a feedback for me to the PFAS analysis. And with that, I would like to pass the time back to the host, and thank you so much for everyone's attention. Maraming salamat po.
Thank you, Dr. Jackie, for that powerful overview of technologies we can use for PFAS biosurveillance. Also appreciate the discussion of high resolution mass spectrometry in the context of PFAS. And now we will have a short 5-minute break. For our dear participants, you can use that time to enter your questions in the question and answer box in your screen.
Victor, I cannot hear you again. No. I think after you play the video.
Can you hear me?
Yes, now
Yeah.
yes.
Okay. Thank you. Oh, okay. Now back to the program. We'll now open the floor for questions. And just a reminder to our dear participants, you can address your questions to Dr. Dimson, Dr. Jackie, or to both. So let's start. So, does anyone have any questions? This is specifically for Dr. Jackie. So, are Waters columns also compatible with Agilent systems?
Well, thank you so much for liking the best column in the market. Waters column is the best, yes. And why wouldn't you want to use it with the best LC and MS in the market too? Because the short answer is yes, we can use our columns in every other vendor's instrument. But I would like to also highlight that our instrument is really one of the best, especially when it comes to PFAS analysis. Contamination kit, that PFAS kit is only available with Waters. So I would highly recommend you to use the best column with the best LC and MS. Thank you.
Okay. So I have actually this is a personal question for me. So to our dear participants, please enter your questions into the field. So I have a question for Dr. Dimson. So coming from perspective of doing your research abroad on PFAS, what are the gaps currently here in the Philippines on PFAS research?
Well, in terms of the PFAS research, we are way behind. So we are just sometimes we're just repeating what were done before, and we just say in the Philippine context, something like that. But actually, in terms of the research, they were already done somewhere else. So maybe the information that we needed, why we have to do this, is because we don't have some information yet here in the Philippines. And we don't know the extent of PFAS contamination, etc. Yeah, but uh, aside from that, we need to be also doing research that wherein the main research question really comes from us and from our settings. So that's where I guess the non-targeted approach to PFAS problem should come in. Yeah. So we are still in the targeted. Maybe because of course we don't have regulations yet for all those legacy PFASs. But something must move in the end. Yeah.
Thank you for that wonderful question, Dr. Ian. So again, to our dear audience, please ask your questions. Okay. So while the audience is waiting for I was waiting for the questions from the audience. So I have another question for the both of you. So should I as a researcher start doing PFAS research through high resolution mass spec analysis untargeted profiling here in the Philippines? So let's say water and air bodies here. So what will be the biggest problems that I will encounter?
Dr. Ian, would you like to go first?
So should I answer first? Okay. Yeah. So I think the main problem will be because now it will not be easy to say that I want to start the non-targeted analysis here in the Philippines for PFAS. It's not easy to say that. You have to specify because PFAS is a big class of compounds. We have to specify ranges. So, for example, you want PFAS polymers or PFAS the novel PFAS but short molecules, etc. Because there's an appropriate technology for that. Do you want a very high resolution mass spec and so on, or just the balance between high resolution sensitivity and the time and so on. So all of these things must be taken into consideration. And in terms of your question, so in that case, we have lots of different mass specs here in the Philippines. And you can collaborate depending on your on the type of question that you want, because each institute has its own mass spec with its own capability. But I think that's the most important, to have a variety of different mass spectrometers available. Even with DESI, for example, new techniques or MALDI imaging and so on. So to have a variety means to have a variety of research as well.
Yeah, so I would like to add on to the point about the scientific question. I think every research comes with the question that we are having in mind. So let's say if we are trying to establish the PFAS amount in Philippines waters, that is usually we are doing regulated, targeted, known PFAS, right? Then in that case, a TQ is sufficient. And we already have a lot of TQs in Philippines. You can, of course, collaborate that way, but always you can buy a new one. Yeah, call me. So, but then if you are trying to look at certain things, so when it comes to targeted compounds, even right now, US EPA 1633, they are only monitoring 40 targeted PFAS out of the whole category of 6,000 compounds. So it may seem very small, but it will really help us to establish a baseline of what is the PFAS situation like in Philippines. But at the same time, if you are contributing to the world's discovery of unknown PFAS, because like different people are creating different compounds, and over time, you will have a different localization of PFAS all over the world. So there may be compounds which are unique to the Philippines. And that's where the discovery work will make it important. Just like with discovering flora and fauna, we are just discovering PFAS, which we don't want in Philippines, but it's unique to the region, then high resolution MS will be required. So it really comes back to the question. But the answer is waters.
Thank you for your wonderful answers. And now we have a question from one anonymous attendee. So, are there already PFAS monitoring activities with local government regulatory agencies such as EMB or DNR? So, this is for Dr. Dimson. But my question for Dr. Jackie is, how did the US and Japan, I know they have recently established a PFAS, a monitoring and regulatory policy. So, how do you think they did it, and how can they be an example for us na what we the Philippines still don't have any policies on PFAS regulation?
So maybe I will go first while the question is still fresh. The first question I forgot already. So anyway, Dr. Ian later he will repeat. I will answer for this question first about let's say, creating a regulation. So what the government has been doing, especially with our US counterpart, we work closely with various government labs in US and we try to create methods. So just like in Philippines, there are so many agencies, everybody is creating a different method. But along the way, we improve by adding more compounds and then we try to harmonize. We want to make sure that it comes with the same kind of sample prep. We want to have the same kind of the method. But because every lab started at a different point, and that's why we have so many different methods. Even for US EPA alone, I think we have 5 different methods. ASTM, there are 6 or 7. And then EU, we have 2 different methods when it comes to just PFAS. So because this is all changing very quickly right now, there will be a lot of time to harmonize all these methods together. So there is no one good fit for all method right now. And if Philippines is interested to create such a method, instead of while we can be parallelly harmonizing with the global team, the global international agencies like ISO and so on, locally, these kind of methods can always be developed as well. And we have the know-how. We will be happy to develop such regulations with the local agencies. Thank you.
As to researchers, PFAS researchers in the Philippines, we are starting to have more research now on targeted PFAS analysis for the sake of just detecting the legacy PFAS in different water and environmental matrices, also fish, food packaging, etc. So those are targeted PFAS research. And this, we hope, can be converted into regulations because these are informations that we can feed to our government. And speaking of regulations, which I also agree with Dr. Jackie earlier, and also I will add to what, as an answer to the earlier questions kanina, there's a need for risk assessments. So because risk assessment is not just it's not just about the toxicity of a certain chemical. It should be the toxicity as applied to exposure and hazards, and it's the entire thing. And the exposure is different in different countries. So that's why the risk will be different here in the Philippines. And also, even the sources of this PFAS, for example, as food, you have to consider how many times are you eating a certain food that contains PFAS and factor that in the calculation of the risk and so on. So, and there are even also risk assessment studies. I think it was from UPLB, based on computational tools. So I think earlier there was mention in one question to Dr. Lacerna about computational tools for risk assessments and so on. So there are small groups doing some parts. The only missing thing perhaps is how to harmonize so that we can be even more efficient in drawing the picture for the Philippines in terms of the PFAS situation.
Thank you, Doc Ian and Doc Jackie. So we need a PFAS consortium in the Philippines. Okay, so for our next question, this is for Doc Jackie. Among your products, what is the most cost-effective for initial screening and semi-quantification of PFAS in water?
Right. I was telling you waters is the solution to go. We don't have this solution, but I'm still gonna tell you the answer anyway. So we have this it's not from waters, but there's this technology in the industry which was mentioned in Dr. Ian's talk, which is known as the combustion IC or also the pyrolysis IC. That is an instrument that is a rather cost-effective version for initial screening and also semi-quantification of PFAS. But you know it is semi, right? So why not do the full one? And that's where you will find us waters. I hope this answers the question.
Thank you, Doc Jackie. And next question for waters again. So using waters technology, what is the amount of time that we are looking at from sample preparation to data analysis to identify PFAS?
This is a question because when it comes to sample preparation, we have this SPE kit. So it depends on whether you are doing gravity drip or you are putting pressure. So that will vary significantly from there. And if there is automation, basically time is negligible because we can just leave it there to do the analysis. But if you are doing gravity drip, it will take forever. So in average, I think I would say maybe around 1 hour per sample. just on average for in case you want an answer.
I guess that question is also applicable to Dr. Ian. So at least in your laboratory, what is the amount of time that we're looking at from sample prep to data analysis for PFAS identification, quantification?
Okay, so there are two questions there. Identification will take years because it's non-targeted. For PFAS quantification, it's only a matter of automation, of course, as Dr. Jackie has mentioned. Since we are not automated in our laboratory, we still use the usual SPE, but from waters. So so that means we have some sort of uh, if we have many samples, then I think it will be 2 days of continuous work from sample preparation to analysis. It's not really always, of course, very difficult to do the targeted analysis. If you have already the method, what is more difficult is the method development and validation part. So wherein you have to do a lot of performance characteristics. But when you have already the validated method, if you have lots of sample, then automation will really be helpful. If you have only a few samples, maybe it's okay to have manual workflow.
Thank you, Doc Jackie, Doc Ian. So we still actually have until 3:50 for the question and answer. So to our dear participants, feel free to type in your questions.
So meanwhile, maybe I will address earlier on there was a open question which I think wasn't answered. So I typed in the answer to the anonymous attendee directly. So this is related to what Dr. Ian mentioned also. The question was about trace level of analysis for very low concentration metabolites in clinical or biological sample. It's not a problem generally when we already know what is the target NRM. So as long as a method has been developed, you have validated, you know the calibration curve, but metabolomics usually you don't have the standards. So at least as long as we know what is the target MRM, we can quite easily analyze the compounds because TQ technology, LCMS and MES especially, is designed for trace level quantitation. So maybe it will be difficult for discovery, but usually what we do is we can always look out for certain journal articles and find the relevant analytical method, including the MRM transitions. So this will be sufficient for any trace level quantitation. Just to share with the rest of the team.
Thank you, Dr. Jackie. And so while waiting for questions from the participants, so I have another questions. So how different will be the PFAS analysis from water and air samples?
Water and air samples, you said? Okay. Yeah. Well, there are two things that we must consider. There are totally a different set of PFAS that would want to stay in water, and another set of PFAS that will volatilize and be in the air. But for one, most PFASs are hydrophobic, so they don't like water. They want to go somewhere else. But there's another issue, the volatility of those compounds. Now, if you want to analyze PFAS in the air, you use either active samplers or passive samplers, and then extract. So for passive samplers, you have polyurethane discs, etc. And then active samplers, you have pumps pass through SPE or other adsorbent materials and then desorb and analyze. Usually this PFAS in the air are analyzed using GCMS. So unlike in water wherein you have but same techniques as well. Yeah. A different maybe a way of calculating or of validating, especially percent recoveries in the air, because now you have something that is volatile. How do you introduce something that is volatile and control their concentration so that you can still use it for your recovery calculation? So those things must be also taken into consideration.
Agree. So let me just advertise for a little because like Dr. Ian has mentioned very clearly, volatility is the key towards the GC side of analysis. And out of the 6,000 compounds under the PFAS category, there are less than maybe around 2%, which are just the volatile ones. So typically the PFAS are longer chain, but recently indeed this short chain PFAS is becoming a very hot topic in the other side of the world. So there has been a lot of concern about analyzing this. But of course, considering the throughput for gaseous volatile PFAS, it's also in terms of investment may be quite challenging to just get a GCMS to analyze these compounds. So that's where I want to advertise about APGC. So APGC allows us to perform GC analysis on the LCMS. So LCMS handles 98% of the PFAS that you normally would found in water or other matrices. But then the other 2%, when we switch to the GC mode, we can analyze it as well. So really, we have that full set of solution for PFAS analysis. Thank you.
Thank you for those wonderful answers. Okay. Are there participants? We still have 10 minutes for a question and answer. We have two experts here, especially those who would want to go to PFAS research. Okay. Okay, so I'm going to ask another question. So, personally, what's the most difficult thing that you encountered in your research with PFAS?
For me, it's contamination. Really, we are using too many things recently. I mean, in our daily lives that contains PFAS. We might even not even think about them, but dry fit shirts have PFAS because that's how we repel the water. We have non-stick coating on cooking pans. We have the plastic pipettes. We have everything. So the contamination from environment is really the biggest challenge about PFAS analysis.
Yeah, I agree with the contamination. That's one of the challenges, physical challenges. So I think you need to have a clean room. You must devote an entire room for PFAS analysis, and where you would keep the samples as well. Uh, However, if you do, for example, the non targeted approach to PFAS analysis, that is very challenging. Despite of course, now we have the software. For example, the water software will make everything fast in terms of the initial calculation. But then putting them together is a different thing. So, and that is where you really have to also spend a lot of effort because you will have information about mass defects. You will have information about the fragments, etc. But how do they fit together to form the molecule, for example? Is a molecule only one molecule, or is it a polymeric mixture of different molecular weights and so on? So that's the challenge, the most challenging part in uh, non-targeted analysis, putting together everything to come up with one structure.
Thank you for those answers. And now we have another question from an anonymous attendee. So, is there any highly specific microorganism that can show PFAS degradation potential aside from water?
Parang I think meron na recently na report. I just have to review on that. Uh, or maybe it's a more general plastics. Yeah. So it's more of now trying to look for organisms that can break the CF bonds. I think I have read somewhere, but it must. I need to confirm. Yeah. But the thing is, it's only at the starting point. Uh,
Okay, so addendum to that question. So what do you think should be our direction as mankind in tackling PFAS degradation? Because it will be a bigger problem in the future, PFAS. So we should address it definitely.
so the approach, as I mentioned in my presentation earlier, uh, when we have information, we can have specific responses. So, for example, if these groups are present, etc. But for now, our responses should focus on those that we can control, and that means focusing on those who are manufacturing the substances to at least let's say, give us details of what their products are. Uh, Not really the public, but at least some sort of the government access to the kind of molecules that they are producing. In Europe, they have uh, the restriction for chemical reach. And the European Chemical Agency that manages the production of chemicals in the European Union. But here in Asia, we don't have, or even just ASEAN, or just in the Philippines, we have some that regulate the polymers, etc. So the industry. But they are not really too focused on PFAS because PFAS is not yet within had reached that level of attention, uh, like in Europe or in the US. So in the US, they are also closely working with the industry. So even those limits for PPT, for the top four PFAS molecules, these limits were reached based on dialogues with different sectors. So in order to have, because now you cannot really lower the limit for the drinking water if the others cannot clean their drinking water. So so that means preventing them to have water, so and so on. So it's really is just for now starting with the things that we can control, especially the government, with what the government can control.
Okay, we have time for one last question. Anyone? Okay, so we have another question. So from an anonymous attendee, uh, do you have suggestions for detection of PFAS?
Yeah, maybe I can answer this. So whether it is livestock or aquatic animals, there have been regulations out there, and we already have published several application notes with regards to those kind of sample metrics. So we are mainly looking at US EPA 1633 and also the EURL method, which is 2022/1431. So these are the methods which have been particularly looking at the regulation of PFAS in food-related matrices. Maybe I'll just type it in the group chat so you can remember the numbers. There you go.
Yeah, so I get this wraps up our question and answer. So we've had a very, very productive discussion. I thank Dr. Jackie and Dr. Ian for participating in this webinar, and to our dear participants. I know I've learned a lot. So and thank you for your thoughtful questions and active participation. Again, if you have any more questions, feel free to type it down, and the resource speaker to whom it is addressed to, we will send this to our speakers. And now, to formally thank our speakers, will now present their certificates of appreciation. So just to read the certificate again, each certificate recognizes their contribution to today's theme, LCMS Metabolomics in Motion, Techniques and Tools for PFAS Biosurveillance, held this November 14th at the Philippine Genome Center, UP Diliman. Signed by Dr. Neil Andrew D. Bascos, our Program Director of the Protein Proteomics and Metabolomics Facility, and Dr. Wendell L. Rivera, our Executive Director of the Philippine Genome Center.
Thank you, Dr. Jackie.
Yeah. So that was our program director, Dr. Neil Andrew Vasquez. So thank you, Doc Ian. So let's just have a short photo. Okay, 1, 2, 3.
Andiyan is Dr. Ian. Thank you, thank you.
Yeah. Okay. And next for Dr. Jackie. Okay, let's just have a short photo. 1, 2, 3. Okay. And now let's take another photo with both Doc Jackie and Doc Ian. Okay. 1, 2, 3. Yes. And again, this concludes our webinar. Thank you for joining LCMS Metabolomics in Motion, Techniques and Tools for PFAS Biosurveillance, presented by the PGC PPMF and co-presented by Waters Corporation and Rainphil Incorporated.